Hi everybody, this is Adam Kinkaya, and I am presenting a presentation called MetaSoft and ForrestPMPlot, a visualization of GWAS meta-analysis. Basically, I'll be taking some genetic data from the GWAS catalog and performing a simple meta-analysis on it using some free software called MetaSoft, and then visualizing the results on a plot using some free software called ForrestPMPlot. So I will do a quick introduction to what is known as GWAS on the GWAS catalog, and a quick introduction to the concept of meta-analysis for heterogeneous studies, basically how we're able to combine results across different studies to get a single composite result that we can use. As I said, I'll be using two freeware pieces of software, MetaSoft and ForrestPMPlot. And as an example, I will be looking at some COVID-19 genetic variants and their potential impact on developing disease. So, as I said, GWAS is the Genome-Wide Association Studies. This is a relatively new technology, the last 10 or so years, that allows us to do genetic sequencing in the entire genome at one time. It allows us to identify potentially thousands of genetic variants associated on a single genome. These are specifically what are called as SNPs, SNPs, single nucleotide polymorphisms. These are genetic variants where the genetic code is a single letter off. So in the genetic code, we would be expecting a C, and instead we would get a T. Or expecting an A, and instead we get a G. So there's a single letter off, a single nucleotide off. These SNPs also would be referred to as variants, can be associated with physical traits in a person or any being. These could be genetic traits like height or eye color or things like that, or they could also be disease related, development of disease or being susceptible to a disease. So, for example, there's a website called the GWAS catalog that publishes millions of pieces of data related to these SNPs that are collected from GWAS. And you can look up genetic traits, physical traits or genetic traits. So, example, for COVID-19, you can put in COVID-19 and get a list of several hundred variants that are believed to be related to the development of symptoms in COVID-19. It could be good or bad. It could make a person more susceptible to development of disease or more resistant to development of disease. If you notice here, this is a few entries from the GWAS catalog for COVID-19. We have 3 different variants here. We have some numeric data, the beta, the odds ratios, the p-value, and then also if you notice on the far right hand side, these are 2 different studies. So we have studies ending in 48 and 49. So these are 2 different studies, 3 different variants. And what we're particularly interested in here is the beta value, which is the unit contribution towards the risk development of a disease. So if a variant is present and it has a beta of 2.35, and you have a single copy of that variant in your genetic code, that means you would have a increase of risk for developing this trait, which is on here, requiring COVID respiratory support. You would have a 2.35 increase in risk. Now, that is not a percentage. That is a number that you would sum up all the risk values from all your contributions of variants on your code. You would sum up all those values to get an overall genetic risk for this particular trait. But this would be the unit increase for this one particular variant, the beta. So that is something we're going to look at coming up. So as I said, meta-analysis, we have multiple studies for the same variants. So we would have a variant that's being detected and reported in multiple studies conducted under different conditions, different populations that are being looked at, different racial demographics, age, conditions, and different environmental conditions. So the question is, so we have results from multiple studies for the same variant, and we get different results for each study. How do we combine all those together to get a hopefully more accurate picture of that we can go forward with a single value that combines the information from all these different studies conducted under different conditions, and how can we do that? Well, one way we can do this is through what is known as random effects modeling, which is a method for controlling the unobserved heterogeneity. So basically controlling for contributions to the model that are not related to the genetic code, things such as demographics, environment, other things not correlated with any particular variable. So we want to do a random effects modeling. We're going to take the beta and the standard error for the beta for each variant and each study. So we're going to have multiple betas and standard errors for each variant, depending upon the number of studies that variant is. So if we have 5 different studies for one variant, then we would have 5 different betas with 5 different standard errors. And then the output of our model will be combining those together to get a single beta for that variant along with a single standard error. And then also what is called an M value. And M value will be used later on. But what's important is here that we're taking the multiple betas and combining them into a single value that we can use. As I said before, beta is the unit increase or decrease in the risk of developing a disease, depending upon if the variant is present or not. The M value that we're going to talk about is the posterior probability. This is the probability that we calculate after we consider the random effects modeling. So it is our posterior probability, means it's calculated after the model is done. And just a little quick math here. In the GWAS catalog, if you notice, it gives us the beta. It does not give us the standard error for the beta, but it does give us the confidence interval, an upper and lower bound for the beta. And we can do a little math here to get the standard error just by taking the upper bound of the confidence interval and doing a little math here, and we can get the standard error. So we will need that standard error when we use the MetaSoft software, which takes in an input of a list of variants with the beta and the standard error for each study, and then outputs this list of the variants with a single beta and a single standard error. So basically, we take all that data from the GWAS catalog, we do a little formatting and putting it into the format that MetaSoft expects, which is each row is a variant, and each column, each pair of columns will be a different study. So we have here, as an example, we have a list of variants, and we have 7 different studies across the top of the columns here. And for each study, we have the beta and the standard error. And you'll notice that there's a mixture here. Some studies, some variants are only on 2 studies or 1 study. Some are on 3 or more. This is just a small selection of the variants. But as you can see, the idea is we have results across the board from 7 different studies, different combinations of variants. Variants are present or not present in certain studies, etc. So once we get our meta-analysis output, as I mentioned, we're going to get a composite beta and standard error. And then I mentioned also we're going to get what is called an M value. And an M value is another way of looking at the significance of a SNP to see how much contribution it would have towards a disease, if any. But this ForrestPMPlot tool, we will take our output from the MetaSoft, we'll put it into this ForrestPMPlot, and it will generate a PM plot such as this example one here. And depending upon the location on the plot, we can make some predictions on the significance of that particular SNP, if we think it's going to have a significant impact on the disease or not. So, as an example, looking at COVID-19, when we look in the GWAS catalog under COVID-19, we get back over 750 genetic variants. These are all related to COVID-19, different aspects of COVID-19, systematic versus non-systematic, requiring respiratory support. It's basically whether you're going to have a severe reaction to a COVID-19 infection or not. That's the trait that is being measured in different ways. So as I said before, we have the beta value and our confidence interval from which we can calculate the standard error. And so we take these over 750 variants from the GWAS catalog, and we need to filter it some and format it to be able to put it into the MetaSoft. One thing is, because we're doing a meta-analysis, we only want to look at variants that are being reported across multiple studies, at least 3 studies in this case. When we do that, it actually significantly reduces the number of variants. There's only 39 variants across 7 studies that each variant appears in at least 3 studies. So we've reduced our list down to 39 variants, from which we have the beta and the confidence interval, so that we can then calculate the standard error for each of those betas of each variant in each study. And then we put all that into a file. We run the MetaSoft with the default settings, which is using the random effects. And this gives us an output file of the composite beta values and that M value. So we have our output file now for every variant. We have our composite beta and the M value. And the ForrestPMPlot, it looks at plots on an individual basis. So we would select one variant at a time from that list to look at the contribution. So there's one variant that actually appears in 4 studies. And so that's the one I decided to look at for ForrestPMPlot. So this is the variant here at the top, RS142891837. We have our original p-values and our original beta, and then the MetaSoft meta values. So you can see the original beta range from 0.72 to 1.06, while the updated composite is about 0.82. Nothing surprising there. What is really interesting on this plot is that you can see the M value are all extremely high. They're all above 0.9, which, if you remember back on the previous slide, it would show us the area on the right-hand side of the plot of the M value PM plot the M values are significant. So what's that saying is that generally the p value is what we would look at for significance in this case. And I know I didn't mention p values because I don't want to get too much into the math, but generally p values are what we look at for significance. And generally a p-value needs to be at least 10 to the negative 8. Anything larger than that is not considered significant. So we would look at this data and they're all 10 to the negative 6, 10 to the negative 7. On a glance, we would probably say, oh, all right, well, this is not significant. We shouldn't look at this SNP. But when you do the meta-analysis, when you take into consideration all of these studies and the environmental and other contributions, we end up with an M value that tells us it's predicted to be very significant. So perhaps the M value is more useful to us in that each individual study's p-value may not be significant, but they're for reasons that have to do with the individual study, the way they conducted the study, and not because of the actual significance, genetic contribution of this variant, which the M value indicates to us is very high. So what this would, from my limited knowledge of this, I would say this genetic variant is likely, very likely to be significant, have a significant contribution towards the development of COVID-19 symptoms. And if you're a doctor and you want to look at your patient's genetic variants to try to give some prediction of what they would experience having COVID, this would be a genetic variant you could look at and say, hey, my patient has 5 copies of this variant, therefore they have 5 times this beta's contributions to the risk. Maybe they should be more mindful of their health when it comes to COVID. So thank you. As I said, this is a very simple example of meta-analysis using data that's already available for free on the internet in the GWAS catalog. Using free software, you can get MetaSoft and the PMPlot tool, and you can make some useful predictions just using this free stuff. So it's pretty cool. All right, thank you, everybody.